In a major genetic study, over 5,000 variants linked to increased cancer risk were identified in the BAP1 gene, revealing the potential for new targeted therapies and improving genetic testing for diverse populations. Researchers have pinpointed specific variants in a gene that significantly raise the likelihood of developing various cancer types. This discovery has the potential to enhance early detection and facilitate personalized treatments for diverse populations. Researchers from the Wellcome Sanger Institute and their collaborators at the Institute of Cancer Research, London, and the University of Cambridge assessed the health impact of all possible genetic changes in the tumor protection gene BAP1. They found around a fifth of these possible changes were pathogenic, significantly increasing the risk of developing cancers of the eye, lung lining, brain, skin, and kidney. The findings, published in Nature Genetics, are freely available so that they can be immediately used by doctors to help diagnose patients and choose the most effective therapies for them. Importantly, as all possible variants were assessed, the findings benefit individuals from diverse ethnic backgrounds who have historically been underrepresented in genetics research. The team also uncovered a link between certain disrupted BAP1 variants and higher levels of IGF-1, a hormone and growth factor. This discovery opens the door to developing new drugs that could inhibit these harmful effects, potentially slowing down or preventing the progression of certain cancers. The BAP1 protein acts as a powerful tumor suppressor in the body, protecting against cancers of the eye, lung lining, brain, skin, and kidney. Inherited variants that disrupt the protein can increase a person's lifetime risk of developing these cancers by up to 50%, typically occurring around middle age. Detecting these variants early through genetic screening can guide preventative measures, greatly enhance treatment effectiveness, and improve the quality of life for individuals affected. However, until now, there has been limited understanding of which specific genetic changes in BAP1 to look out for, especially for rare variants that cause it to malfunction and fuel cancer growth. The team also discovered that people with harmful BAP1 variants have elevated levels of IGF-1 in their blood, a hormone linked to both cancer growth and brain development. Even individuals without cancer showed these elevated levels, suggesting that IGF-1 could be a target for new treatments to slow down or prevent certain cancers. Further analysis revealed harmful BAP1 variants and higher IGF-1 levels were linked to worse outcomes in uveal melanoma patients, highlighting the potential for IGF-1 inhibitors in cancer therapy. Notably, the technique profiles all possible BAP1 variants from diverse populations, not only those prevalent in European clinical records, helping to address the underrepresentation of non-European populations in genetic studies. Previous approaches for studying how variants affect function in genes have been on a very small scale or exclude important contexts that may contribute to how they behave. This new approach provides a true picture of gene behavior, enabling larger and more complex studies of genetic variation. This opens up new possibilities for understanding how these changes drive disease. This research could mean more accurate interpretation of genetic tests, earlier diagnoses, and improved outcomes for patients and their families. Scientists want to ensure that life-saving genetic insights are accessible and relevant to all people, regardless of their ancestry. Our aim is to apply this technique to a wider range of genes, potentially covering the entire human genome in the next decade with the Atlas of Variant Effects. Thank you for watching. If you've enjoyed our videos, subscribe to our channel, for there's more to come.